Hello everyone, my name is Pankaj Bhaumik. I'm a senior research scientist and team lead, Advanced Plant Biotechnology team at the National Research Council Canada, located in Saskatoon. I'm also the scientific support lead for NRC's Sustainable Protein Production Program, where we work very closely with the industry partner, academia, non-for-profit, and other government organizations. First, I would like to thank Janelle and the organizing committee from Pulse Canada for the opportunity to participate as a speaker in this virtual workshop addressing pulse flavor challenges. We all know that one of the barriers to increased use of pulse products in the food industry is the off flavor, and the pulse flavor issues continue to plague the food industry and pose a barrier to increased use of pea and other pulses in new food products. But before I present our recent progress about the breeding and genetic approaches to improve flavor, I would like to give you a brief overview of the NRC and our Sustainable Protein Production Program. Those who are not familiar with the National Research Council, it is Canada's the largest federal research and development organization. We have three primary roles, which includes advancing scientific and technical knowledge, supporting business innovation, as well as government policy. We have access to more than 2,000 scientists, technicians, and other specialists who undertake the research for NRC, and they are working in roughly 200 facilities across Canada. So I mentioned about the Sustainable Protein Production Program. As a scientific support lead, I'm working for this program, over 5 years now. And our vision for this program is to increase the value of plant-based proteins and their co-products, moving them up the value chain. And how do we do it? We collaborate with the Protein Industry Canada Supercluster and pursue research to help Canadian industry address existing challenges through sustainable production practices as well as innovations in processing and manufacturing. Furthermore, our program's mandate aligns with governments of Canada priorities, including climate change, food security, and economic growth. I mentioned about the program. So what are the key focus areas of the Sustainable Protein Production Program? So we have three main focus areas. As you see here, the first one is the improved protein sources. And that's basically the objective is developing new genetics and new protein crop varieties as a source material for processing, processability, or quality attributes. Second one, the sustainable production practices for reducing the chemical inputs through precision agriculture as well as bio-based solution, such as use of inoculants. And finally, the third area, processing and manufacturing innovations, includes protein processing itself as well as valorizing the co-products. Now I'm coming to our team. I mentioned that as a team lead, Advanced Plant Biotechnology team, our key focus areas, we work, with the NRC, the platforms, other platforms to provide solutions for Canadian and global partners. We have genetic engineering, plant tissue cultures, we have strong analytical skills, and we bring extensive experience in rapid germplasm improvements for increased climate resiliency, crop productivity, and enhancement of the quality traits. Now I'm coming to the projects, that funded by the Saskatchewan Ministry of Agriculture, the P off flavor projects. When we started that, through the program, Sustainable Protein Production Program, we wanted to first look at, what are the genetic basis, or latest biotechnology tools that's already there. So we did a review paper, and published that, I think around 2022, where the comprehensive review showed us that there are already different tools in terms of genetic engineering, or also the pathways for different traits. Here, as you see, the different genes and for controlling different traits already have been studied in legume crops. We also looked at the existing examples of using CRISPR-Cas9 or novel plant breeding tools in other legumes, and we published that work as well. and we found that, in terms of the successful story, soybean, the model legumes, even the chickpea, there are examples of applying CRISPR-Cas9 mediated gene editing to improve the legume crops. So on the basis of that, we started several projects, like our ADF-funded P lipoxygenase projects. We also started to work on chickpeas, lentil, grass peas for different traits, and anti-nutritionals to address not only improving the protein or yield, also, for example, drought tolerant, Um, but today, I would like to focus updating you on our progress in terms of the successful gene editing in peas as a proof of concept. YOLO-P, identifying the uh, off flavor or reducing off flavor for ingredient application. So why did we, select YOLO-P in this case? We all know that, Canada's pulse industry is booming and exceeding 1 billion annually, and Canada is the world's leading exporter of peas and lentils. This makes pea improvement a significant economic opportunity. Secondly, a substantial portion, around 75% of the Canadian pea market is YOLO-P. However, we know that there are challenges. Many pea products suffer from the beany flavors or off flavors or undesired flavors that affect taste and consumer acceptance. Therefore, we targeted the lipoxygenase and the off flavor as a trait for this project. And when we looked at the available literature, John Innes Center's, their mutant population on peas already identified two lipoxygenase genes in peas that are contributing to the off flavor. So we targeted both LOX2 and LOX3 to create the null mutant as an option for creating variation and to look at whether that really translate in the reduction of the enzyme activity, translate the reduction of fatty acid profile, the volatiles, and the substrate linoleic and linoleic acid. So as I mentioned, so therefore, we hypothesized, or we focused, as an objective of our ADF-funded project was to, we would like to develop pea lines with reduced off flavor and anti-nutrients. But today we'll be talking only for the flavor aspects. And why do we want to do that? Solve the end-use problems with pulse ingredients and data sets, develop data sets and inform other parts of the value chain, while you are selecting varieties for your specific application for peas. But we had no idea in terms of what's the existing lipoxygenase activity in a population or in the elite breeding germplasm. So we collaborated with CDC, Crop Development Center at the University of Saskatchewan. We received several lines from Tom Orkentine, and we started characterizing for lipoxygenase activity. As you see here, we had some idea that there are some variation, not maybe very significantly different, but CDC Meadow, for an example, here CDC Amarillo, well adapted varieties, they had lower lipoxygenase compared to CDC Spectrum. So that's why we selected CDC Spectrum as a proof of concept, to knock down the lipoxygenase and then compare whether that translate into the off flavor, reduced off flavor and reduced enzyme activity. So this is routine, as I said, we do as a platform here at the NRC Saskatoon. It's crop agnostic. So we have applied our tissue culture and transformation platform in cereals, in oilseeds. So the only difference is in terms of efficiency of tissue culture response of legumes. Legumes are considered as recalcitrant species. So we were playing with different germplasms, modifying our protocols. Still, we are continuing on that to improve the efficiency. So as a routine platform or gene editing, usually what we do, we first design several guide RNAs, usually 5 guide RNAs we design on the basis of the sequence of the specific genotype, in this case CDC spectrum. So we amplified the lipoxygenase from CDC spectrum, sequenced it, and on the basis of a specific sequence, we design guide RNAs, and we test it in vitro first, the cleavage efficiency, and on the basis of the best guide RNA, we do the stable transformations. For example, here you can see guide RNA 1 and guide RNA 3 are selected. We also do using protoplast to confirm whether the guide RNA 1 and guide RNA 3 is creating the edit. So from the sequence information, as you see, there is a deletion of T. We confirm that, and then we move to the stable transformation and regeneration of the plants. This is a standard method, agrobacteria mediated, P transformation that we use. Use this in an embryo axis, sliced embryo axis as the explants, transform it, confirm with gas staining that they are positive, then basically transfer to different tissue culture media for shoot and root induction, then it goes to the soil. But we had problem like very few seeds we get, so we had to take these plants to the greenhouse, advance that we call the T1 and T2, so that not only we can confirm the edits what is inherited in the subsequent generation, at the same time we have enough seeds that will allow us to test for the traits that we are interested. This is again to show you the similar way. Even when we are advancing the lines, we were continuously making sure that the same edits is inherited in T1 and T2 lines, the deletion. So we generated total 17 lines, and here the 3, 4, 5, 14, and 17 that we are showing the sequence showed the inheritance of the sequence. And then there were like homozygous and heterozygous edits that we noticed. So we kept 3 of them we selected, and then before we had enough seeds to do all the characterizations, we have a unique way, the electronic nodes. It's basically chemical sensors, 32 different chemical sensors that we use to screen, to look at whether we really see significant difference among the wild type and the different transgenic lines. Several plants from each line we characterize, and then that gave us the confidence that no, we see the differences. And also we didn't have enough seeds, so we couldn't do the wet chemistry, but using the near infrared spectroscopy, that also came very handy. We also looked at the basic like total protein, carbohydrate, and the fiber content, moisture, ash, to make sure that the proximate analysis of those lines compared to the wild type, we didn't change any significant compositions of the seeds. So that gave us the hope that okay, now if we have enough seeds, we could do the wet chemistry, go to the lab, and then look for the lipoxygenase enzyme activity, fatty acid profile. Here we are just showing several lines we did, only 3 lines we are showing, and how the precursor linoleic acid and linoleic acid results showing the differences compared to the wild type. So as you see here in the wild type and the transgenic lines, the enzyme activity, wild type 170 unit per milligram, and we have 3 different. We couldn't do complete knockdown, that's the reason, because lipoxygenase also contribute to your ABA and your initial germinations and other physiological traits that we have to test in the field. So we wanted to have a knockdown, but a range of, not complete knockdown. So you can see that our objective, we were successful creating a range of variations. What our hypothesis was, we'll see unused linoleic and linoleic acid in the transgenic lines because of the low lipoxygenase activity. We also looked at those lines for volatile components. Here we are just showing 2 of the common abundant volatiles, the hexanol and 2-hexanol, that's published according to the published work, is abundant in peas, and probable origin from where the precursor is coming. And as you see, the wild type and the transgenic lines, there is significant reduction in terms of both hexanol and the 2-hexanol. We also did the same data, but showing all the lines. We wanted to show the correlogram, and the partial least square regression, where you can see that our analysis using the correlogram and the partial least square regression showing strong correlations, between lipoxygenase activity, fatty acid composition, and volatile organic compounds. Um, so this statistical modeling, clearly demonstrated that reduced lipoxygenase activity directly translates to fewer off flavors, and these findings, we believe, provide compelling evidence, supporting the effectiveness of our gene editing strategy in improving pea flavor profile. Now what? So we completed our project, 3 years funded project, Saskatchewan, Ministry of Agriculture. So we would like to take this to the field. So we went back to the ADF and successfully we received funding for 2 more years, where we would like to uh, go back to the Canadian Food Inspection Agency with the evidence, with the data from the confined research field trial, that our lines will not uh, be considered as a uh, in terms of novelty identification, will not have any of these changes in endogenous protein, will not increase levels of non-endogenous allergen, and it will not have any impact on key nutritional composition or metabolism. And we didn't intentionally change the food use of the plant, and of course it will not result in the presence of foreign DNA. We have to show that there is no foreign DNA in the final product. And as I mentioned, that we are waiting for the field trial this spring, where the data will generate to address this environmental release novelty determination. As I mentioned, these are the 5 key questions that we have. With our data, we have to prove that this there is no increased potential of the plant, that it will become a weedy species. There is no gene flow from the peas. We know from the biology already that it's self-pollinated, so there will be no gene flow. And importantly, from the herbicide and pathogen and pest, that it will not create any negative impacts for the biodiversity. So this is where we are. While we are waiting, for the confined research field trial, we also looked at the existing, what are the, according to the Canadian Food Inspection Agency, what are the other examples that we have? And we have seen that, the CFIA already listed, several non-novel gene-edited products, including, as you see here, the mustard greens with reduced pungency, potatoes with improved tuber yield, which demonstrate the feasibility and safety of the approach. So these successful examples, pave the way for wider adaptation, we believe, for the gene editing technologies in pea improvement as well. Next few slides, I'll just share some success story from our program, that our team's innovative approach was highlighted as success story, and it demonstrates the potential for gene editing to improve the nutritional value and market appeal of pulses. One thing I would like to clarify, that our approach is not to directly use these lines, but creating this variation and identifying the genetic basis of the trait, whether it's saponin, bitter taste, or off flavor. Similarly, you see the University of Calgary here. We collaborated with the University of Calgary on the saponin reduction projects that I'm not presenting here, focusing on the flavor aspects. But the idea is this line could be used to cross or transfer to the elite line, the trait, or use as a marker for the breeding program. Another example, this is our publication, another example from the website that highlighted, the International Service for the Acquisition of Agri-Biotech Applications, and this organization is a non-for-profit, and it shares the benefit of crop biotechnology to various stakeholders. So they highlighted our work as well. The Western producer also interviewed everyone involved in pulse gene editing, Tom Orkentins, Avin Benija, myself, and they also published an article considering that although the acceptance for the market acceptance, but still the tools and the traits that we are creating variations will be critical for the breeding program. I'm almost at the end of my presentation. So in conclusion, what I would like to highlight, from the update, that what is the problem if the question is that we are trying to solve. We all, have experienced that the unappealing flavors and aromas, those are formed by the ingredients due to the activity of lipoxygenase, and or volatile organic compounds and the fatty acids. So why is this problem important? Because it limits the consumption and utilization of environment-friendly pulses in the food industry. Who is most worried about this problem? We know we already reached out to stakeholders who are worried this problem, including food manufacturers, culinary professionals, consumers, and the pulse industry as a whole. And when will these seeds be tested in real world conditions? So we are hoping that through the confined research field trial coming spring, these lines will be able to test and generate that data. Is anybody in the industry interested? We already have an industry partner supported for the ADF funding, and there is significant interest from the food industry. So what I would like to mention is that through these projects, our research successfully developed the high throughput method for p germplasm screening, gene editing, and we have established the robust protocol for guidance and validation, protoplast transformation, plant transformation. And we believe that this will proceed to the field trials and ingredient testing that to validate that this improvement in the real ingredient applications. We greatly acknowledge the contributions of our research team, including Wei Yang, Brittany Polai, Chami Munashinge, Wei Yang, and Murali Nara. Our success also relies heavily on the valuable collaborations with Drs. Jim House, Michelle Aliani, and many others, as well as the generous funding from ADF, SASPALS, Genome Canada, and other partners.